Welcome to our bioanalytical laboratory located in Seattle, Washington, where ligand-binding assays, flow cytometry, PCR, and mass spectrometry come together under one roof. Here, precision, collaboration, and scientific integrity drive one mission: to unveil the invisible, shaping each molecule. The open concept layout allows for efficient teamwork between our scientists and technical staff. From sample receipt to data generation, each workflow is carefully orchestrated to ensure optimized sample flow and strict adherence to GLP and GCLP standards. Our PCR suites are designed for both digital and quantitative PCR applications, including ddPCR and RT-ddPCR, providing exceptional sensitivity for vector copy number, gene expression, and transcript quantification. These capabilities are essential for gene therapy, cell therapy, and mRNA-based modalities. Within these temperature-controlled environments, each instrument delivers high-precision amplification and quantification of nucleic acids. Scientists can accurately measure transgene persistence, monitor biodistribution, and assess therapeutic response across time points. Our workflows are designed to reduce variability and maximize reproducibility. Whether it's quantifying AAV vector genomes, monitoring immune-related gene expression, or supporting pharmacodynamic assessments, PCR remains one of our most powerful molecular tools. Our flow cytometry lab is where cellular dynamics are brought to light. Through multiplexed analysis, we can characterize immune cell subsets, evaluate cytokine expression, and detect biomarkers that reflect therapeutic efficacy or immune response. High-throughput instruments, such as the A3 flow cytometer, enable precise quantification of multiple cell markers simultaneously across species, matrices, and study types. This allows for comprehensive immunophenotyping and detailed mechanistic insight. With sophisticated gating strategies and data analytics, flow cytometry empowers our scientists to track subtle cellular changes from activation to exhaustion and to correlate these findings with pharmacological outcomes. These data not only guide biomarker discovery, but also inform clinical translation. Together, these capabilities bridge the molecular and cellular worlds, creating a full picture of biological response. The LC-MS bench area is dedicated to sample preparation for quantitative bioanalysis of both small and large molecules. Every step, from extraction to purification, is optimized for precision and reproducibility. Automated workflows and rigorous quality control procedures ensure the integrity of each analytical run, while minimizing human error and maximizing throughput. All biological samples are stored in temperature-monitored freezers ranging from 4 degrees Celsius to minus 80 degrees Celsius, with continuous data logging and alarm monitoring systems for around-the-clock oversight. Redundant power systems and strict inventory tracking safeguard sample integrity at every stage of the study life cycle. Our mass spectrometry suite houses multiple high-sensitivity LC-MS/MS systems dedicated to method development, validation, and sample analysis, facilitating the quantitation of drugs, metabolites, and biomarkers. Each LC-MS/MS instrument is paired with HPLC systems to handle diverse matrices, from plasma and serum to cerebrospinal fluid and tissue homogenates. Dedicated hoods, fume extraction systems, and safety showers ensure full compliance with health and safety regulations. Across all analytical platforms, our teams apply validated methods, well-defined quality systems, and scientific expertise to deliver reliable, regulatory-compliant data that drives smarter decisions. From early discovery through clinical development, our integrated approach, uniting molecular, cellular, and analytical sciences, supports bioanalytical programs of every scale and complexity. Every sample tells a story, Every data point advances understanding. And through it all, we're unveiling the invisible layers of biology that guide tomorrow's therapies.